Welcome to the deep dive, where we take some complex research and really break it down for you.
Yeah, trying to get you up to speed quickly.
Exactly. So today we're diving into something really important and honestly incredibly expensive in the real world.
hmm
How do we actually measure, properly measure the environmental impact when we do huge infrastructure projects?
It's a massive question.
Mm.
And today we're focusing specifically on road repairs, or rather, culvert replacements. You know, those big pipes under roads.
Right, that let streams flow
Yeah.
through.
Yeah. And in Washington State, this isn't just routine maintenance. We're talking about a massive court-ordered program.
Billions of dollars to replace hundreds of these things.
Billions, yeah. It's a huge undertaking.
And the reason behind it is critical. It's about salmon, right? These culverts, if they're not designed well, they block salmon from getting upstream to spawn.
Which destroys their habitat, and importantly, this actually violates treaty rights, sovereign treaty rights of the local indigenous tribes who rely on those salmon runs.
So the stakes are incredibly high. You've got treaty rights, massive costs, critical habitat. You absolutely need to know if replacing these culverts is actually working.
Absolutely, you need proof. And that's where the research we're digging into today comes in. It offers a way to get that proof quantitatively.
Okay, so let's name the source. We're looking at the paper, Quantifying Impacts of an Environmental Intervention Using Environmental DNA. The lead author is Elizabeth N. Allan.
And our goal here is to unpack how they use this really cutting-edge genetic technique, environmental DNA or eDNA,
Right.
to assess these culvert replacements. Because the old ways of doing it, well, they just weren't cutting it.
Yeah, let's talk about that. Why are our traditional environmental impact assessments, or EIAs, often not good enough for something like this?
Well, the problem with traditional methods is often they're based on, literature reviews, maybe some field measurements.
But only for a couple of species, maybe?
Exactly. They're expensive. They take a lot of person power out in the field. And honestly, the monitoring is often, well, lacking.
How lacking are we talking?
There was one review mentioned in our sources looking at stream restoration projects. Get this, 62% of them had zero pre-project monitoring. None at all.
62%. That's staggering. How can you measure impact if you don't even know what it was like before you started?
You can't, really. It makes justifying the expense and the effort incredibly difficult, especially, like you said, with billions of dollars and treaty rights on the line.
Okay, so clearly we need a better tool, something more scalable, more comprehensive. Enter eDNA.
Right. Environmental DNA. This is the genetic material that organisms leave behind. Skin cells, mucus, waste. It all ends up in the water.
So you just take a water sample.
Yep, just a water sample. And using a technique called metabarcoding, you can potentially identify the DNA traces from thousands of species in that single sample. It's like a genetic snapshot of the whole community.
Okay, that's powerful for seeing who is there. But the paper's title mentions quantifying impacts, so just knowing the species list isn't enough, is it? What's the gap metabarcoding alone can't fill?
That's the crucial point. Standard metabarcoding gives you compositional data. It tells you, like, maybe 80% of the fish DNA reads came from cutthroat trout and 20% from coho salmon.
So the proportions, the ratio.
Exactly, the relative abundance after the DNA gets amplified in the lab. But it doesn't tell you the absolute concentration. It doesn't tell you how many actual DNA copies of cutthroat versus coho were in that original liter of stream water.
I see. And you need that absolute number to say definitively, okay, after we replace the culvert, the total amount of salmon DNA upstream actually increased by X amount.
Precisely. You need to go from just ratios to actual quantities to track changes over time reliably. And figuring out how to do that, bridging that gap, is really the core innovation in this paper.
Okay, so how did they tackle that? The methodology must have been pretty intense.
Oh, it was. Very rigorous. They set up an 18-month study, constant sampling.
18 months? Wow.
Yeah, and they used a specific design called an asymmetrical BACI design. That stands for Before-After Control Impact.
Okay, break that down. Before-after makes sense. Control impact makes sense. Why asymmetrical? What does that add?
But think about a stream. Things change all the time, right? Rainfall, temperature, seasons.
Sure, huge natural fluctuations.
Exactly. So if you only look at the stream where you did the work, the impact site, before and after, how do you know if a change you see is because of your work or just because it was a really dry year overall?
Ah, right. You need something to compare it against, the control.
You got it. The asymmetry comes from having multiple control sites. So they monitored the treatment site, Patton Creek, where they replaced two culverts, one under State Route 11 and a much bigger one under I-5. They sampled that monthly for the full 18 months.
Okay.
But, Dest, they also monitored 4 other nearby creeks, Barnes, Chuckanut, Squalicum, and Portage, which had existing culverts of different types. Those were the control sites.
So changes in Padding Creek were compared against the normal ups and downs happening in those other 4 creeks.
Precisely. It lets you filter out the background noise, the seasonal stuff, and isolate the effect of the actual culvert replacement. Super important for making valid conclusions.
Makes sense. Okay, methodology part 2, the lab work. They focused on 4 key salmonids: cutthroat, coho, rainbow steelhead, and sockeye kokanee. How did they get from water samples to quantitative data for all 4?
It was a really clever two-pronged approach. First, they did the standard metabarcoding using a specific genetic marker, the 12S MyVish marker, which is good for identifying lots of fish species. That gave them the proportions, like we discussed.
Okay, step 1, proportions of everything.
Step 2 was the key for quantification. They also ran quantitative PCR, or qPCR. But here's the efficient part. They didn't run qPCR for all 4 target species. They developed a highly specific qPCR test for just one of them, the cutthroat trout.
Just the cutthroat.
Just the cutthroat. That qPCR test gave them the absolute concentration copies per milliliter for that single species.
Okay, hang on. So they have proportions for all 4 salmon from metabarcoding and an absolute number for just one, the cutthroat, from qPCR. How do you connect those? How do you get absolute numbers for the coho, rainbow, and sockeye? That seems like the tricky bit.
It is tricky, and it involved a critical calibration step. This is where it gets quite technical, but also really neat. They use something called mock communities.
Mock communities, like fake fish.
Ah, not quite. More like lab-created DNA cocktails. They mixed known amounts of DNA from different fish species together in precise, known proportions.
Okay, why?
Because when you run metabarcoding, some species' DNA sequences amplify better or more easily than others in the PCR machine. It's called amplification bias.
So the raw proportions you get out might not perfectly match the proportions that were actually in the water.
Exactly. If cutthroat DNA amplifies twice as easily as coho DNA, your results will look skewed. The mock communities, because you know the true starting proportions, let you build a correction factor. You see how much bias there is for each species and mathematically correct for it.
That's smart. So you adjust the metabarcoding ratios to reflect the true ratios.
Right, and applying this mock community correction to eDNA from water samples was actually a novel part of this study. It hadn't really been done this way before in this context.
Okay, so now they have bias-corrected proportions for all 4 salmonids.
Yep. And now they can use that absolute concentration number they got for the cutthroat trout from the qPCR. They basically anchor the corrected proportions of all 4 species to that known cutthroat quantity.
I get it. So if the corrected data says coho is half as abundant as cutthroat, and they know the absolute cutthroat number is, say, 1,000 copies per ml, then the coho number must be 500 copies per ml.
You've got it. It allows them to estimate the absolute concentrations for all 4 target species by only running one expensive, highly specific qPCR assay.
That is a huge efficiency gain. Imagine trying to develop and validate separate qPCR assays for every single species you might care about in every river system.
It's exactly. This combo approach, broad metabarcoding plus one targeted qPCR anchor, makes quantitative monitoring much more feasible at scale. But there was one more crucial step.
Oh, what else?
They had to account for the water flow. Stream discharge.
Right, because a high concentration in a tiny trickle of water is very different from the same concentration in a raging flood.
Precisely. Dilution effects are massive. So just reporting DNA copies per milliliter isn't enough for comparing across seasons or different flow conditions.
So what did they do?
They measured the stream discharge, how much water was flowing past per second at the same time they took the eDNA samples. Then they multiply the DNA concentration, copies per liter, by the discharge, liters per second.
Okay, concentration times flow rate.
Yeah.
That gives you.
Copies per second. It's an eDNA mass flow rate. It tells you the total amount of DNA passing that point over time, regardless of whether the stream is high or low. It removed the dilution effect and makes the measurements truly comparable. Figure 2 in the paper shows this really well.
That sounds incredibly rigorous. That step seems vital for making the data defensible, legally or for policy.
Absolutely. It isolates the biological signal from the sort of hydrological noise. Okay, so they have this robust, flow-corrected quantitative data for 4 salmonids across 18 months and 5 creeks.
And they analyzed it all using a linear mixed effects model, which helps statistically separate the intervention effect from all the other variables like site differences and seasonality.
Correct. That model lets them ask, After accounting for everything else, did the culvert replacement at Padden Creek actually change the salmonid eDNA flow rate compared to the control creeks?
So let's get to the results. What did they find about the existing culverts before the replacements? Did the eDNA confirm they were barriers?
It did in some cases. Barnes Creek, one of the controls, showed a really strong barrier effect. They only found salmonid eDNA upstream of its culvert in 3 out of 12 sampling months.
Wow, okay, so fish basically couldn't get past that one most of the time.
Pretty much. And the big I-5 culvert on Padden Creek also showed clear signs of being a barrier before it was replaced. The eDNA signal was consistently lower upstream compared to downstream.
But I sense a but coming. Were there surprises?
Yes, and this is really interesting from a management perspective. The data suggested that some culverts that had been officially flagged as barriers might not have been as bad as thought.
Really? Like which ones?
Well, for instance, the other culvert on Padden Creek, the one under SR11, didn't seem to be blocking fish nearly as much as the I-5 one, based on the eDNA patterns.
Huh. So this method could actually save money by helping prioritize. It could tell the state, hey, maybe focus on the I-5 culvert first. The SR11 one isn't as urgent.
Exactly. That's a major potential application, using real-time ecological data like eDNA to guide these incredibly expensive decisions rather than relying solely on older assessments or physical models alone. Huge potential for smarter spending.
Okay, now for the million-dollar, or rather billion-dollar question. What happened during the construction? They replaced these massive structures. They had to dewater the creek. I'd expect a huge negative impact, at least temporarily.
That's what you might think, right?
Yeah.
But this was probably the most significant finding for the managers and the tribes involved. The study found that the construction events themselves, the dewatering, the work, the rewatering, had only transient and fairly minor effects on the salmonid eDNA signals.
Minor. Really? How minor?
The ups and downs they saw in Padden Creek during the construction periods were actually pretty similar in size to the natural fluctuations they were seeing in the control creeks over that same time frame.
Wow. So the disturbance from the actual replacement work was basically lost in the background noise of natural variability.
Largely, yes. It suggests that if the construction is managed carefully, and they did things like installing temporary fish passage routes during the work, the short-term negative impacts on these key species can be minimal.
That's huge. It provides justification, doesn't it? It suggests you can do this disruptive work to get the long-term habitat benefits without causing a major short-term crisis for the fish populations.
It really does. It supports the idea that the long-term gain outweighs the short-term pain, which was a major concern.
Did they learn anything else? Maybe about when the barriers were worst?
Yeah, they did see a relationship with flow. The biggest difference between upstream and downstream eDNA signals the clearest sign of a barrier effect tended to happen during the summer months when the water flow was lowest.
Okay, that makes intuitive sense. Harder for fish to push through a culvert when there's barely any water.
Exactly. It confirms that the barrier effect isn't static. It gets worse under low flow conditions.
And the eDNA did pick up known seasonal patterns too, right? Like migrations?
Oh yeah. For example, the signal for sockeye and kokanee salmon peaks strongly in Padden Creek between November and February, which lines up perfectly with when those fish are known to run in that system.
But we should mention the nuance there. eDNA is great, but it can't tell apart, say, a resident rainbow trout that lives there all year versus a migrating steelhead trout coming in from the ocean because genetically they're the same species.
That's a key limitation, absolutely. The eDNA signal represents the species, but not necessarily the life history stage or whether it's a resident or migratory individual. It's kind of a smoothed out biological signal integrated over the area upstream.
Smoothed out, is that a bad thing? Does it limit the usefulness?
Well, it depends what you're asking. If you need an exact count of fish passing a specific point on a specific day, eDNA isn't the tool. Traditional methods like traps might be better for that, though they have their own biases.
Right.
But if your goal is long-term environmental impact assessment, understanding the overall presence, relative abundance, and changes in the fish community over seasons or years in response to an intervention, then that smoothing is actually a strength because it integrates the signal over space and time. It's less susceptible to the massive day-to-day, spot-to-spot variability you get with, say, electrofishing. A single net haul can vary wildly depending on exactly where and when you do it. eDNA gives you a more stable, representative picture of the system's overall biological state over your sampling period.
Okay, I see. So for tracking the success of a multi-year restoration project, That integrated signal might actually be more reliable than a few snapshot counts.
Exactly. It's arguably better suited for assessing these broad-scale, long-term intervention effects.
So zooming out, what are the big implications here? This seems like more than just a study about salmon in Washington.
Oh, much bigger. This provides a template, a scientifically robust and quantitative framework for how environmental impact assessments could be done worldwide, especially where they're mandated by law. It moves beyond subjective assessments or simple presence-absence.
And it's efficient too, right? That combo of one qPCR plus metabarcoding.
Huge efficiency gain. That makes quantitative monitoring practical for potentially hundreds of sites, not just a few intensively studied research locations. Think about scaling this up for regulatory monitoring across a whole state or region.
It really changes the game for accountability.
It does, though of course we need to acknowledge limitations and next steps.
Sure, like the resident versus migrant issue we mentioned.
Right. And also, this study covered 18 months, capturing the construction phase. The real proof of success will come from longer-term monitoring.
We need to see if fish populations truly rebound upstream over the next, say, 5 or 10 years, right?
Exactly. Future work needs to continue monitoring these sites to document that sustained recovery and confirm that the barriers are truly gone for good, not just during the construction phase. And maybe look at a wider range of species, too, beyond just salmonids.
Makes sense. So the main takeaway for me is that we finally have a tool, this quantitative eDNA approach, that seems up to the task of rigorously monitoring these massive, expensive environmental interventions. It moves us beyond just hoping for the best.
It provides that crucial link between spending billions on restoration and actually proving it worked, a blueprint for accountability.
Okay, so let's leave our listeners with a final thought. If this quantitative eDNA method can give us reliable, hard data on the impact of fixing salmon culverts, what other huge human activities could we, or maybe should we, be monitoring this way?
Yeah, think about dam removals, huge impacts, huge debates, or massive dredging projects in harbors, or large-scale wetland restorations, all multi-million or billion-dollar efforts.
Imagine having this level of quantitative insight into whether those projects are delivering the ecological benefits we hope for.
The potential for transparency, for adaptive management, for just knowing what works.
Yeah.
It's enormous.
So the question for you listening is, what major environmental change, what big human intervention would you want to see monitored with this kind of powerful quantitative eDNA approach?
Something to think about.
Definitely. Until next time on The Deep Dive, keep digging deeper.